We're back to a fully virtual format this week for the HNRCA Monday seminar series. We have another exciting seminar planned for today. So the format for today's seminar will be our invited speaker presentation, followed by time for questions. Please use the Q&A box in Zoom for your questions, which you are welcome to submit at any time during the talk. Our speaker today is Dr. Michael Snyder. He is the Stanford Asherman Professor in Genetics at Stanford Medicine and Chair of the Department of Genetics. He is also the Director of the Center of Genomics and Personalized Medicine. Dr. Snyder has pioneered the use of big data and multi-omics to advance scientific discovery and transform healthcare. His laboratory has invented many technologies that are widely used in medicine and research, including methods for characterizing genomes and their products. His research includes application of omics and wearable technologies, and he has co-led large-scale projects, including ENCODE, Human Microbiome Project, Human Biomolecular Atlas Program, and Human Tumor Atlas Network. So for our audience here involved in research on nutrition and healthy aging, characterization of these omes, the genome, transcriptome, metabolome, is essentially important and expanding research area. And I'm even say it's becoming an essential part when studying variation with respect to disease or biomarkers of disease and aging. So we are very interested in your research. Thank you for taking the time to speak with us today. I'm now pleased to introduce our speaker, Dr. Michael Snyder.
All right, well, great. Thanks so much for having me here. And I think I know some of you already through various programs, and so some of this may be a little redundant, but hopefully you'll learn some new things as we go along. So many of you may know, and as I was introduced very generously, that our stick is really to try and use big data to transform healthcare. I do think the healthcare system's broken in many, many different ways. Some of the key ones that bother me are shown on this slide. It's very focused on sick care, very reactive. When people are healthy, we typically measure very few things. Also, when you're healthy, you don't get measured very often. Most people don't go to the physician's office. And the biggest one that I find bothersome is this one down here. Nearly every decision about your health is based on population-based measurements. Instead, I think we would all agree it's best to be focused on keeping people healthy. As such, be very proactive. We're certainly capable of measuring many, many more things than we currently do. The frequency, I would argue, should depend on what you're at risk for, what your trajectories look like. And lastly, I would think we'd all agree again that we should be focused on individual-based healthcare rather than population-based. Physicians say we do this now, but I would argue not at the level that it could be. And so overall, we should be focused on precision health rather than precision medicine. And so just to illustrate this point, I like to show this example because it kind of goes counter to what many people think, which is that you've been told since you're little that your oral temperature, excuse me, I guess that's why these seminars are virtual, so that you don't have to worry about me spreading germs. I actually don't think I'm sick, I have allergies, but anyway, sorry, so back to the point. You've been told since you're little, your oral temperature is 98.6, but if you actually look at the data that's out there, it's really more like 97.5 for median, and this is just one of a number of studies. I myself have 97.3, but more importantly, there's a spread. This is a 25th quartile, 94.6, and this is a 75th quartile, 99.1. So what that means in today's world, if you go to a physician's office, they put a thermometer in your mouth, they'll measure you at 94 point, and they measure you at 98.6, and your baseline is normally down here, say 94.6. You're actually up 4 degrees. They'll tell you you're normal because you're 98.6, but you're actually up 4 degrees over your baseline. And so you're clearly probably not normal. Something's off if you're 4 degrees above that. So that is a big theme of our work. Know what your baseline is so you can detect these shifts. So I think we all know that our health is influenced by many things. It's influenced by your genome, your DNA, but all these other things as well, which is very pertinent to this particular group: exercise and activity, food, stress, and various exposures, environmental, pathogenic exposures, all influence your health. We know this intuitively, but we're now in a world where you can measure this. You can now sequence a genome for probably pretty soon for $100 with some of the new technologies coming out. Food is clunky, but we can score that. Activity is a lot easier to score, and so on and so forth. We can measure these. And even if we have trouble measuring them, we can measure their effects by basically doing deep molecular physiological profiles on people. But so many of you may know, a number of years ago, we set up something we call personal omics profiling, where the idea is we take deep data dives on people. We basically sequence their genome once, and then out of their blood components, we'll measure their epigenome, meaning their DNA methylome, transcriptome, proteome. These are all measured out of peripheral blood monocyte cells, your immune cells. We also measure proteome, cytokines, now 80 cytokines, growth factors, metabolome, lipidome. These we measure out of plasma. Microbiome, we do the gut and nasal, though we've more recently done tongue and skin. That turns out to be very, very interesting. This is on top of deep clinical testing, questionnaires like stress questionnaires. And then we had advanced tests like stress echocardiograms, and I'm type 2 diabetic, so we have a very strong emphasis on glucose control measurements, things like oral glucose tolerance tests and such. And then a number of years ago, before the Apple Watch came out, we started bringing wearables because we realized they're powerful. I'll talk about that today. Have a big program there. And so one aspect is this deep data dive on people. And then the other aspect is longitudinal. So we'll sample people every 3 months while they're healthy. And then if an adverse event comes along, like a viral infection, we'll usually take 5 to 7 additional samplings. And so we've been running this on a smallish cohort, 109 people. I'm one of them. I've been doing me for almost 13 years now. But most of the cohort, we've been doing around 9.5 years, some a little longer, some a little less. So you might say, well, why are we doing all this? Well, we're trying to understand what it means to be healthy. How does it change over time? How do healthy profiles differ between different people? What happens at the earliest times of illness? And one interesting aspect from the medical side is can these deep data dives, these new technologies that we use that we're embedding here, can they be used for better managing people's health? And with regards to that last point, can we use genome sequencing, et cetera, to manage health? The answer is yes. It turns out from the first 109 people, 49 had a major health discovery, and it spanned a wide range of areas. So hem-oncology, cardiovascular disease, metabolic disease, infectious. All these areas we found some things that turned out to be pretty important. And I want to emphasize these were all found pre-symptomatically, meaning these things popped up because we were doing these deep data dives on people. And I like to think of it as if your health is a 1,000-piece jigsaw puzzle. The way we do it today is you measure 7 or 8 pieces. And what we're trying to do is measure more like 800, so you can get a much clearer picture of what your health looks like. And so, and some of these turned out to be a pretty big deal. So we caught some with early lymphoma, 2 people with pre-cancers, 2 people with serious heart issues. And there was no one technology found this. Sometimes it was gene sequencing, sometimes it was wearables, imaging. Often it was multiple assays were important for actually seeing something was off. And again, I want to emphasize this is all found pre-symptomatically. So just to walk you through a few examples, genome sequencing was very, very powerful. From the first 70, we've done more now. The numbers are still roughly this. But from first 70 people, 12 had a single gene mutation that put them at high risk for certain kinds of diseases. The classic, these are called Mendelian diseases. The classic is BRCA, where a single gene mutation BRCA puts women at high risk for breast and ovarian cancer. And so one of our individuals did have that. 3 of the people had something that turned out to be pretty important. They're probably all important. They all get screened as a consequence of this. But this individual, fairly young, had a mutation here. It turns out this is in a cardiomyopathy gene. And this turns out his father died of a heart attack shortly after he sequenced him. His aunt had one in her 50s. And then he did a follow-up. And what you know, he actually has a heart defect. So we think this is the culprit. And so he's on medication as a consequence. One of the individuals has a mutation here. There's that 2X means 2, I should say 2 did. But one did a whole body MRI follow-up. They were at risk for certain kinds of cancers because of this mutation. Sure enough, they had early thyroid cancer. That part was removed and they were able to maintain most of their thyroid. 9 individuals coming in the study were thought to be diabetic. It turns out that One of them they were all thought to be type 2, but one of them really has a mutation in this gene. It's a MODY gene, and the treatment for a MODY person is a little bit different from type 2 diabetes typically. And so they've actually shifted. They're on better medication now as a consequence. So even though it's a minority of people, 12 out of 70, if you're one of these 12, it's a big deal. And then one person learned a pretty important pharmacogenomic variant as well. So this is the Mendelian disease. I won't have time to get into this, but we're spending a lot of time on these complex diseases like polygenic diseases they're thought to be. These are thought to be due to common mutations of low effect. And the way people are scoring these now is with something called polygenic risk scores, where what you do is you typically sum over thousands, if not millions, of these single nucleotide polymorphisms and make predictions about, your risk for this. And it works for the top couple percent. It worked for me in type 2 diabetes, believe it or not. It's actually predicted from my genome by a tool Butte. And so it does work, but again, you can look at the data behind this. It really only works for the top 2 or 3%. And I think the reasons for that is severalfold. One is that it assumes genetics is linear, and anyone who does genetics knows it's not. You can't just sum over mutations. It's rarely linear, actually. If you have 2 mutations in the same pathway, 1 + 1 is 1. 2 mutations in separate pathways, 1 + 1 can be 15. And so it's not linear at all. And not only that, it ignores all the rare variants. So we've come up with new ways, and I won't have time to get into this. We're actually trying to define, if you will, the genetic basis of a lot of these complex diseases and uses machine learning. It's turned out to be very, very powerful. We have 2 different approaches. We're about to marry them into one. But for this first one, abdominal aortic aneurysm, we took rare variants. We said using machine learning, give us all the pathogenic mutations in cases relative to controls for a fairly small cohort, 401 people were sequenced. And we went from what was 3 genes known to 60 genes and found a lot of genes. We could even set up a predictive model for this, for who will get the disease. That's pretty decent. We also have now gone on to actually using GWAS signals and mapping open chromatin regions. I'll encourage you to look at these papers. We won't have time, but we can actually, using machine learning, this is turning out to be really transformative. From the open chromatin region, you can look for those that are rich for GWAS signals. And for ALS, we went from 7 known genes to 690 and went on to validate using iPS cells for one of the genes and such on. So it's actually turned out to be very, very powerful. And more recently, we've done this for severe COVID, like when young people get COVID and wind up in a hospital. That's thought to be due to genetics, or at least influence. And from a very, very large GWAS study, we went to 47 genes over 1,000. And we could actually even define based on where the enrichment for open chromatids. It turns out it's natural killers cells, and T cells are the ones that are most likely for why you'll get severe COVID. And so again, it won't have time, but we think this is going to be very, very transformative for getting genetic basis disease. Okay, that's just one sort of technology that's there, genome sequencing, but other technologies have been powerful. We caught someone with early lymphoma. That was because they had an enlarged spleen, as well as several blood markers that were up. We had 2 people with these precancers that can convert to aggressive cancer. So one person, also fairly young, had way more IgM than everybody else. And so we thought something was off. They default, sure enough, there are way too many IgM producing cells that can convert to aggressive cancer. There's another one called smoldering myeloma that's actually even more severe. Same kind of thing. They were shifted, in that case, an IgG. So again, these deep data profiles do reveal things. 6 people had plaques in their arteries. I'm one of the 6. They're relatively modest, but they're still there. Mine is not me, but it looks like this. So these, smallish plaques. And recommendation here is increase your statins, which was done for me. I shifted to these PCSK9 inhibitors, and boy, do they work. They really, really drive your cholesterol way down. Many of you may know that. So my plaque is shrinking as a consequence. We're very interested in type 2 diabetes. I'm sure you know that this is an endemic, worse than the pandemic, in my opinion. So coming into the study, as I said, 9 people thought they were diabetic. That's this group here. But when we actually measure them, 2 people were diabetic and didn't know it. And of course, a ton of people are pre-diabetic and didn't know it. That's very common. 9 out of 10 pre-diabetics don't know it. Now, we're very interested in how do people become diabetic? Do they just gradually get there? Does something trigger it? What's going on? And so it turns out during the course of the study from the first, it turns out 4 years, 9 other individuals became diabetic and others had diabetic range values. And then others became pre-diabetic as well. So we can look at these and say, well, for those who became clinically diagnosed as diabetic, there were 9 of them. How did they get there? And it turns out that 7 of the 9 just gradually got there, slowly went up. And this is one of them. This person went up in their fasting glucose shown here in orange, or their hemoglobin A1C, to the point they became diabetic and there was an intervention and they got it somewhat under control. This person got there through oral glucose tolerance tests. We think these things tease out different things. And 2 of the people actually of the 7 gained weight. So that was probably the culprit. The other, as a means of this happening, the other 7 or the other 5 didn't gain weight. So that they just gradually got there. It's not clear the mechanism. Interestingly, though, 2 of the 9 individuals actually got there by as though they spiked there. And I'm one of the 2. This is me, 8 years of data as shown here. And I first became diabetic after a very nasty respiratory syncytial virus infection that basically I was going in for glucose, insulin sensitivity tests. I was actually insulin sensitive, you'll see in a minute. But it turns out that while I was in for that test, my fasting glucose was high, my hemoglobin A1C was high, and then got retested again later. And of course, I went past, I was officially diagnosed as diabetic. Now, I know this doesn't look like much, but I basically, that's 10 months of data. I shifted after getting diagnosed, I actually cut out all sugars, I doubled my biking, started running, and I brought my my glucose levels back to normal range. And I know that looks like a tiny blip, but that's actually about 10 months to a year of being very elevated. But I was at least initially able to get it under control. So this, as I say, was predicted from my genome. And so what we think is that it's a combination of my genetic predisposition along with the viral infections, what probably triggered this. So my glucose was running boringly low. And even though I was collecting data, I kind of stopped looking at it. And once you know, somebody else looked at it here and said, Mike, you realize your hemoglobin A1C is back to 7.0 and later got to 7.1. And I said, no, I had no idea. By the way, 6.5 is when you're officially classified as diabetic. So we looked back and sure enough, it had spiked up again. And 2 things happened here. One is I did get this a second viral infection, but the other is I actually had stopped running. So it's not 100% clear what's going on. I did start running again. I brought it down. I never got it all the way down. So it was somewhat elevated. And I basically kept up the running. I was running about 4 times a week, 4 to 5 miles a run, about as much as I thought my schedule could handle. It just gradually kept creeping up. But so finally, I decided I'll try something else. I shifted to weightlifting because muscle mass is better for glucose homeostasis. I did gain 10 pounds of muscle mass. I do whole body MRI, follow that, but it didn't work. I just kept creeping up. So finally, I bit the bullet. I went on metformin, and when I was in the high 6s here, and it turns out I'm a metformin non-responder, even though I kept increasing the dose. I went all the way to 7.5. And so what was the solution for me? Well, the answer is more data. It turns out we measured other things, including glucose disposition. And as I mentioned, I'm insulin sensitive. I actually make insulin just fine. We could show that by other means, but I don't dispense it very well. And it turns out then there's a drug for that, rapinolide, and I respond to that quite well. These days, I'm on an SGLT2 inhibitor, which also works pretty well for me. The only minus with that is that it does lead, in my case, I'm pretty sure, leading to loss of muscle mass. So something I was not trying to do. I kept up the weightlifting. So who wants to lose what muscle mass after that? Anyway, the point out of all this is that we call this precision diabetes. By being able to better understand what's wrong with someone, we can come up with the right, in this case, drugs. I'm still trying to use diet to get this under control. So I've been playing with some things you'll see about in a minute. All right. One thing that was kind of interesting is this was the first time someone had shown that type 2 diabetes could be triggered by a viral infection. It's been suggested by type 1 previously, but not for type 2. And of course, many of you may know this is really turning out to be rampant, somewhere on the order of 2 to 4%, probably more like 4% of individuals who get COVID wind up diabetic. It's often not discerned whether it's type 1 or type 2. Many cases it is type 1, but it's pretty clear both are on the rise when if you get a COVID infection. So our data first suggests this. And I won't have time to show you this, but what was kind of interesting in my case is actually what we saw was that my methylome shifted in advance of my glucose going on. So after those viral infections, my methylome shifted. It's very, very interesting. And it's right upstream in front of my metabolic genes. Not a big shift, but it's across hundreds of genes. That's, again, kind of a cool result. Right. One of the most important things we learned from all this profiling is that, well, as we're profiling healthy people, we discovered that we all have a distinct profile, and it's very, very stable. So this is just showing 4 of the omes. It's true for all the omes, microbiome will be there as well. And I'm just showing 12 people with 10 healthy visits. I have 6 years of data here. And what you'll notice is that I'm the light blue. Every one of my visits, these are the dots, actually cluster near each other. And you see the brown persons are together. I'm the light blue. Here's the red. Here, you can't see that so well. There's yellow. Point out of all this is that we have these, profiles. And again, they're very personal. They're very robust. They're very stable. And so not only that, it turns out if you get a viral infection, or in this case, we had people run to their VO2 max and we sampled them after the run. And you will shift your profile. In this case, for the exercise, you shift your profile such that 50% of your molecules actually shift significantly, but you still look more like you than anyone else. Even me, who did this thing twice at the beginning and end of study, my dots still piled up on top of each other. Some were VO2 max and some were baseline. And again, so these, what that means is that the difference between the perturbed you, whether it's viral infection, exercise, what have you, and the healthy you is closer than the difference between a perturbed you and someone else. And so the reason that's a big deal is that if I want to see if you're getting ill, so to your purpose of seminar, it's a lot easier if I can just compare a time point from you now to your healthy baseline and see if you shifted, than it is to try and compare a time point for you now to 500 other healthy people's, because the differences between people is different more than the difference between a disease and a healthy state. For the most part, there are exceptions. There are biomarkers that you can try and follow too. But in general, what I say is true. So that, again, that means you really want to get your healthy profile now while you're healthy so you can detect these shifts. And this has really popped up a lot in our IPOP. People are still in the healthy range, but had shifted from their baseline. Let us help catch disease early. So there's a lot of other fun things you can do with these data when you start collecting big data around people. One of the fun things was to sort of see how many biological seasons there are. So I told you most people have stable, healthy patterns, but there are shifts, and there are 2 shifts that we've studied. One is seasonal and the other is aging. So it turns out, because we're collecting samples throughout the year at high frequency, we can, from the many tens of thousands of molecules and microbes we're following, we can see which ones have seasonal patterns. And it turns out there's 1,192. Oh, so backing up, for the seasonal one, we wanted to just see how many biological seasons are there. You've been told since you were little, there are 4: winter, spring, summer, fall. But, and they're, they're labeled as 3 months each. And if you think about it, who says? Maybe there's 15, maybe there's 3. And why would they be 3 months each? That doesn't even make any sense. That's totally arbitrary. So we wanted to let the biological data tell us. And so what we found was that from these tens of thousands of molecules and microbes, we found 1,192 that show a seasonal pattern. Some were known, but we actually mapped them at pretty high resolution, which wasn't known, but most weren't known, of course. And we have microbes and all kinds of interesting molecules. So we can then say, all right, we have all these patterns. How many do they fall into? And the answer is 2. We can show this mathematically. There are 2 major patterns. You say, well, that makes sense. These people they're from Northern California. One will be winter, one will be summer. And you would be half right. Here's the winter pattern. These are molecules that peak in January, go down, and they come back up. And it's the stuff you would expect, viral infections and things associated with that. Acne, that's known, believe it or not, to have peak during those periods. But the other one's not summer at all. It turns out it's late spring, early summer. It's April, May, late April, May. And there, in hindsight, it makes sense. Allergy and asthma molecules, eosinophils, for example, are here. But there's other stuff there that in hindsight makes sense. Cardiovascular disease, metabolic disease, type 2 diabetes markers, they also peak here. And our interpretation of this is that even in California, where, it gets cooler in winter and rains, people don't exercise as much. And so these markers build up, and then it gets warmer, and then people, become more active, and then they go back down. There are other things in this pattern I don't understand. Molecules associated with seizures and sleep peak then as well. And so trying to figure out what that means as well. So what's also interesting about this, I don't want much time for this, but there are clinical markers associated with these patterns like creatinine and things like that. And so it's worth taking this information into account when you're thinking about people's health and how you would relate that, meaning if it's coming April or May and people's numbers are high, you might realize they'll probably get back down as summer comes along and so on. So the other time that people's patterns change is over time, the other periods, and we interpret this as aging. And the nice thing is, because we're sampling people reasonably densely, we can see how people are aging, and it turns out everyone's aging differently. This one's me, 4 years of data. My coagulation, metabolic pathway, they all go up, which is pretty typical. And this just shows my coagulation pathway. Here's another person, their top pathway that went up is actually a cardiac hypertrophic signaling pathway, meaning they're a cardio ager. That's their top pathway that shifts. So we think, by the way, you need as few as 5 samplings within 2 years, and we can see how people are aging. And so we've gone on to take all the people, we had 43 people with enough data, and we asked how many patterns are there? And the answer is we found 4 statistically significant patterns. I know there are more than 4, but we're not empowered to be significant for some of them, like the cardio, where we only had 1 example. And here are the 4: kidney, liver, metabolic, and immune. And we call these ageotypes. These are aging phenotypes that we can see at a molecular level. And each column is a different person. Each row is an ageotype. And so the people over here, they're aging in all 4 categories. This person's aging in 3 of the 4. I'm somewhere in here. I'm not much of an immune ager, but I age a lot more for the others. Kidney, this person, kidney and metabolics and so on. So we can see how you're aging. And once again, this information is actionable. There are markers associated with these ageotypes, and I'll just show you, here's 6 of the markers. Here's an easy one to interpret: hemoglobin A1C, the marker of type 2 diabetes. For the group as a whole, it goes up. Every one of these is an individual, and the dark lines means they're statistically significant. So these 4 people did go up, just like the group as a whole. But these other 4 people actually had theirs go down, and we can associate that with lifestyle changes. So like 2 of them went on a diet, 2 of them lost weight, 1 exercised. And so we could see these shifts. And again, that's a no-brainer pretty easily. But there are other people like this creatinine, a marker of kidney function. The group as a whole went up, but 10 people significantly went down. And interestingly, 8 of the 10 are on statins. So there may be some interesting interactions there that's worth pursuing. And so this is a big push for us now to correlate lifestyle changes with these metabolic markers. And again, I think it's very, very relevant to this particular meeting group. So one thing I get asked a lot is how you're going to scale this. And I'm in Silicon Valley. I'm a believer that academics aren't good at scaling. I think it's what companies do well. So we formed a company called QBio, totally conflicted here. I forgot to mention this. Everything I'm telling you is conflicted in this talk. And so this company, just in the first 100 plus people, it's way more than this now, but you can see they had, we caught some pretty early stuff. So in many cases found multiple lines of evidence. That's what I mean by multivariate. So we caught some of the early ovarian, prostate. We even caught some of the early pancreatic cancer, which is almost never found. And that was found through longitudinal profile. We saw shifts, cardiovascular disease, so on and so forth. So again, these are a big deal. I forgot to mention that both our IPOP cohort and this group, they tend to be older people, 53-ish at the time of enrollment. So, the chance of finding something are higher probably than if you profile 25-year-olds or something. But nonetheless, we can see these shifts. And so, again, this is how we plan to scale this. So another thing I wanted to tell you about, since the nutrition group, and I presented this maybe to some folks in the Boston area nutrition, so this may be redundant for you, but we're doing a lot of studies now around nutrition, quite a few. And this is just one of them around the topic of fiber. So, you've been told since you were little, again, especially now, fiber is good for you. It sits in your colon, supports your microbiome, releases polyphenols, boosts your immune system, all these good benefits listed here, heart disease and so on and so forth. The thing is, fibers are very heterogeneous, like saying all animals are the same. It's long chain, short chain, hydrophobic, hydrophilic, positive, negative. It's all over the map. And so again, there's no such thing as one fiber. So we decided, maybe because my chemistry training, we would come in one fiber at a time and see their effects. And we started with two most popular ones, inulin and arabinoxylan, which is found in Metamucil, and this is various sources. And the literature is all over the map on these things. They've both been reported to have positive effects on glucose, as well as cholesterol. It depends what study you look at. So we looked for ourselves with our deep omics profiling. What we did, it's a small group, but it's a crossover study. So you really learn a ton from this, where we put 18 participants through two different fibers, this arabinoxylan or inulin. They're randomized in the first one. Then they go through a washout, switch to the next one, go through another washout. We put them on a mixture of those two fibers plus three others. And what we do is we do a ramp up. We start them out with 10 grams per day supplement, then 20, then 30 for each of these fiber periods. And we'll sample very, very densely, the beginning, first week, second, third, and during the washout, and so on. So we're trying to see the effects of these fibers on people's molecular profile and see what's going on. And so we do as I say, our usual ohm, the 6 ohms that I mentioned before, along with some deep clinical labs, and to see again what happens to the effects of these fibers. And what we discovered is that this is the arabinoxylan. They both will cause big shifts in your molecular components. So here we go in a ramp up. These are the things that respond to arabinoxylan, go up. These are the ones that go down and then return during the washout and so on. So these are the sorts of things, again, that we think are being driven by the fiber. There are other patterns as well. So it turns out, to get to the clinical side of things first, arabinoxylan does decrease cholesterol in the group as a whole. So meaning, and quite significantly, you can see arabinoxylan driving cholesterol down in a dose-dependent fashion. Interesting, the mixture, which has a fraction of, the total of the arabinoxylan, I think it's 2 grams initially, does have an effect, but not as much. So it's almost dose dependent. Inulin had no effect on the group as a whole. We actually learned other things when we saw these shifts. It turns out that some of the bioacids also shift. They didn't shift with inulin, but they shifted with the arabinoxylan. We could actually see primary and secondary bioacids increase. And so we also saw other changes as well. And I'll explain all this in a minute, but we saw microbiome shifts as well. So these are the microbiota that shift. There are different kinds here that will shift in response to inulin. We could see which ones, again, change. These are the ones that shifted in the presence of arabinoxylan, just at the highest time points, but it's generally dose dependent. And these shift to both. So some fibers cause shift in a very species specific fashion. And so from the shifts that we saw, as well as some of the proteomics work, I won't have time to show you all this, but we could come up with a new model for how we think arabinoxylan shifts cholesterol levels. And the prevailing one, and that's so possible, is that cholesterol directly binds to fiber and gets arabinoxylan specifically and gets swept out. But our data actually says it's probably metabolized through these bioacids. And we can see some inductions of some receptors, as well as particular microbe shifts that lead to this more revised model, that this whole metabolic pathway for how cholesterol is actually metabolized and shifts, and it does involve the microbiome in our model. One thing I thought was kind of cool that we discovered, I told you about population averages in general, but there are exceptions. And what I want to show you is that here's a response to arabinoxylan. This individual, number 69, is a typical example, goes down, comes back up in the presence of a ravenous eyeland. But there's another individual who actually doesn't respond to ravenous eyeland at all. Yet it turns out that person goes down in the presence of inulin, even through some of the initial washouts. And this person is typical, doesn't respond. So the point is that people do respond differently to different fibers, and it probably reflects some of this thing we need to figure out, whether it's due to the epigenome, whether it's due to the microbiome or what have you. There's no question microbiome has some play in this. Aaron Siegel's lab and others have shown this, Justin Sonnenberg, but it still only explains a fraction of the variance. We want to try and figure out the whole thing. All right. I should have time to tell you a little bit about some of our wearable work. I might go through this kind of quickly and summarize some of it. A little bit behind, but we're very keen on wearables because instead of doing these static measurements, now we can actually do follow people 24/7 with wearable devices. And we first got into this before the Apple Watch was out. We really wanted to see, whether we could use these for tracking people's health. We realized they're more than just fitness trackers. They're actually, which is how they were used at the time. They're really health monitors. And they're powerful because they make hundreds of thousands of measurements on you every day, even the cheapest device. So the best devices make 2.5 million measurements on you. And now it was originally said 20% of the US. Now the numbers shifted to 25% of the US is wearing some sort of smartwatch or ring. It's quite a few people. And they're powerful because they measure lots of different things as well. Heart rate variability, they're all accurate for that. Respiration rate, some measure SpO2 and blood pressure. I don't think they're that accurate on the absolute level, but they can find shifts just fine. And some of them have accurate skin temperature devices, some not so much. And so the way you use these, you probably know, is you figure out people's circadian patterns and weekly, and actually weekday versus weekend patterns, that sort of thing. And you can track, all these different activities, so sleep, heart rate, skin temperature. And it turns out, this is my pattern. I sleep at night, wake during day, occasional nap, back sleep. Heart rate slow at night, high during day, back down. Skin temperature, which is different from oral temperature, usually high, drops, comes back up. And so we all have these patterns, and we all have different baseline and measurements for heart rate, blood oxygen, what have you. And then the name of the game is to detect shifts. And what we did, I'll skip over this a little bit and get to the newer stuff, but what we discovered early on is you can tell when people are getting infectious disease because their resting heart rate jumps up. And we first figured this out when I got my Lyme disease. I picked it up asymptomatically because actually my heart rate shifted and my blood oxygen dropped as well. And I later learned my skin temperature dropped. And we went on to show that actually I had gotten Lyme. I did get symptoms later, and I was treated by it with doxycycline, which cleared it up. But when I got tested, sure enough, I was Lyme positive. It's a really well-controlled experiment, as it turns out, because I'd given blood right before I was on a trip. And bottom line is I had seroconverted after getting Lyme disease. We can again follow all that and so on. So we went on to, once we first figured out for Lyme disease, I had 2 years of data where I was wearing a smartwatch. And it turns out I was ill 4 times during that period. And here they are here, viral infection here. This is now we know a viral infection. There's a 4th time as asymptomatic. Here's the Lyme time. This is low resolution, but here's resting heart rate and skin temperature. Every single time I was ill, we saw the shift in resting heart rate and skin temperature. And so we actually published this some time ago, including this part. We actually wrote a detection algorithm for following this. We call it change of heart. Worked well for heart rate, didn't work well for skin temperature. Can get into why that is, but this is it with it. So you do a sliding window, you figure out people's baseline, look for a shift. And sure enough, it worked on me. Every single time I got ill, you could see the shift up in resting heart rate. It's a delta plot. You'll see it in advance of symptoms indicated by the red line. Worked on me, worked on 3 other people, one of whom got sick twice. So the point is, every single time someone got ill, we could see this shift in resting heart rate. Well, along came COVID, as you know. So we published that now almost 6 years ago. And we're improving the algorithms, building an infrastructure. It's pretty cool, I'll tell you about. And so along comes COVID. We're still getting something like 60,000 cases reported each day. It's way more than that. People start reporting this stuff. And so we're still, this is here to stay. This is what we're living with. And if you think about how we measure things now, we use temperature, which is a 300-year-old concept and doesn't work very well for COVID. Or we use, stochastic sampling like antigen tests or PCR, which work, but you don't do them all the time. So we think wearables could be very, very powerful for this, since that's what I'm going to tell you for a few minutes here. So we quickly launched a study once COVID came, and we enrolled 5,300 people, 32 of whom got COVID. And this is our very first case. This is an individual looking at resting heart rate. It's a standard deviation plot. And so here is a diagnosis day. Here is a symptom day. And what you'll notice is that 9.5 days prior to symptom onset, this person had a shifting resting heart rate. You can't miss that. It's very, very clear signal. And this is a real-time detection algorithm I'll tell you about in a minute. And it turns out it worked in 80% of the cases. And the median was this early, we could see this early shift up 4 days prior to symptom onset. Okay, so it worked 80% of the time we'd see it before at the time of symptom onset. So as you might imagine, then we then spent a lot of time building a real-time detection algorithm. It's a cloud-based system, and it's actually scalable to millions and millions of people. So I want you all to sign up so we can keep improving our algorithms. And so right now we have over 10,000, but we want to get we want to get millions. All right. So we have these various detection algorithms. What they do is they build a baseline around your circadian and weekly and monthly patterns. And then we look for abnormal signals from those patterns. It's an anomalous detection algorithm. And we now have some machine learning versions of this that we think will be quite powerful. Then we have a fairly lightweight algorithm, night signal. It doesn't break the bank. We scale this to millions of people. And it turns out it also works. So same number, about 80% of the time, we can detect COVID at or before symptom onset. All right, so so this is one of our first cases again. This person had their symptoms appear here, but they were getting red alerts 3 days prior to symptom onset. So you get either red alert means you got an anomalous signal, green means you don't. And so we, again, red alert symptoms, and they were diagnosed the next day. So we don't get a perfectly stable baseline on everyone. I think getting more data types, that's why we want more people in our study, will be powerful. But it works on both pre-symptomatically as well as asymptomatic case, works on Apple and Fitbit. I know it'll work on all of them because Garmin's high enough resolution, et cetera. So, and the bottom line is here's an asymptomatic case with Fitbit. I could show you with Apple. So here's the diagnosis day, and they were getting red alerts for about 2 weeks prior to symptom onset. So again, we can see all this. I want to emphasize this is not specific for COVID. So anything that shifts your resting heart rates, there's a little bit of steps and a little bit of sleep in the signal. We can give you these red alerts. And it turns out workplace stress is number one. Intense exercise, not just your routine running, but if you run a marathon, your heart rate will be up for a few days, excessive alcohol, so on and so forth. And other infections too, as well. In particular, other infections like mycoplasma, we picked up other things that we can detect. Okay. I can tell you personally, it worked for me. Where we have the alarm set off to go off roughly every 6 weeks or so. And so it turns out that, okay, so it says 5 minutes left. So let's see, I don't know when they sent that, but probably now. All right, so anyway, I'll just tell you that this is my data, a very personal example. I think we can wrap up for 5 minutes. I had a particular family stress. I had to fly out to see my daughter. So that was a red alert day, came back, red alert day. I was getting ready to go to New York City. And I woke up that morning, it was a little bit congested. So is it allergies? Not sure. So I do an antigen test. And sure enough, I come out negative. All right. I look at my smartwatch and I'm a bright positive. Okay. So what do? I got on the plane. Big mistake. I arrive in New York City next morning to go to the meeting. You got to get tested. Wouldn't you know? Bright positive here. So the bottom line, and I therefore had to spend an entire week in New York City in a tiny little hotel room, all because I listened to my antigen test and not my smartwatch. So what this means is that your smartwatch is more sensitive, less specific than an antigen test. And so we need to actually work on that a bit, but you get the idea that we think, again, this is a very, very sensitive assay. 2 beats per minute elevation, you can pick up pretty easily with the smartwatch because you're measuring somebody 24/7. Okay. I'll just go on to say that this is just the beginning. We can pick up other things, for example, stuff associated with anemia, like red blood cell count, hemoglobin. Using machine learning, we can grab features from a smartwatch. This is with a very old smartwatch too. We could actually pick up signals. They're not clinical grade signals, but they're good enough to see these shifts. Even hemoglobin, A1C, and fasting glucose levels, we can pull from a smartwatch. For continuous glucose monitoring, we do a whole bunch here, really a lot. What we did here was we just started putting them on normal and pre-diabetics. This is a time when everybody's using them for type 1 and insulin-dependent type 2. We said, well, let's try them on normal people. And once you know, we discovered that a lot of people are moderate or severe spikers, some just as bad as diabetics. Some normal people spike just as badly as diabetic, normal by fasting glucose, hemoglobin, A1C. And so we wrote algorithms and went on to classify people in the glucotype. So either low, moderate, or high. These are high spikers. These are pretty well controlled. And then we discovered, as did Aaron Siegel, this is Aaron's work, that different people spike to different foods. So you may know this, and some people spike to pasta, others banana, some to bread, some to white bread, some to brown bread, what have you. We're all different in the way we respond to our foods. This work from Aaron's paper is pretty unusual. He had someone who spiked to banana, but didn't spike to a cookie. Probably the only person on the planet doesn't spike to a cookie. This person spikes to, cookie, which is normal, and not a banana. So again, people spike to different foods. Here's a glucose and bread differences and so on. We found that, same thing. People can spike to bread and peanut butter, protein bars, which have carbs in them. We found 80% of people spike to cornflakes and milk. I think that stuff's pretty evil, probably worse than smoking. All right. So anyway, we've gone on with my company, totally conflicted here, but we showed that just wearing a glucose monitor and a simple app, you can actually suppress, you'll improve, I should say, people's time in range, whether healthy or type 2 diabetic. Most people just wearing a monitor for 10 days will show an improvement without really trying very hard. We've run now an intervention trial we'll be publishing showing that actually that you can improve weight and time in range by wearing these monitors for both healthy pre-diabetics and diabetics. So we think this is the future. CGM is a way to scale this to lots and lots of people. All right, so we built an app to pull in all your wearable data, your clinical data, even your microbiome data. We can pull and display in real time, well, semi-real time. This you have to get offline, some of this data. And this is the future I would like to see, where people will get their genome sequenced before birth. And then together with these omics and wearable measurements, we'll be able to better manage people's health for predicting and monitoring people, predicting disease risk, monitoring the health, and catching disease early. So I do want to say we do spin off a lot of companies, and these days we're doing quite a bit in the metabolic health. January AI is an AI CGM company involved in metabolic health. And we have a new one at home testing based on some technology I'll be talking about soon. For remote sampling that lets you actually follow people's metabolic health. And we'll send around links if you want, so you can sign up for these various things if you're interested. And so I'm fortunate to have an amazing lab. Because I'm late, I won't go through this much, but we have Tracy McLaughlin's my clinical collaborator, Jordan Weinstock on the microbiome. This genomics work is, I think, is really quite transformative. It's here. Big wearables team now, all built around trying to detect COVID and disease early using smartwatches and other wearable devices. And so that's the story I wanted to tell you. I think I saved time for some questions, and so happy to take those if you have any.
All right. Thank you, Dr. Snyder, for that excellent presentation. This is really interesting. So with our remaining time, we can take questions. Dr. Snyder, I can read, so we have questions they submit in the Q&A box. I can read the questions out loud for the audience. Is that okay?
Yeah, that sounds good. Why don't you do
that?
That's easiest.
Great. So
sure.
We'll start with a question by Catherine Micton. Oftentimes we hear the phrase, treat patients, not numbers. Do you think the advent of highly individualized, but very data-heavy precision medicine would help or hinder doctors in minding this phrase?
Great question. If we do it right, it's going to help. I view this stuff just like your car, where you have sensors all over it, but you have a dashboard that relays info back. And you can have this dashboard relay to both you and to the physician to be able to better manage people. And I think what we need is just a better way of pulling in data, displaying it. Even now, people don't use longitudinal data properly, even in today's health system. So people don't detect shifts. And that turns out to be very, very important. We've learned from both our research and from some of the companies I've been involved in. You can see when people have that early shift, something's going off, and it's important to catch that. So I think we can bring all this in a monitoring system that physicians can use, patients can use, and it'll be actually the combination of both that really improves their healthcare. So great question.
Okay, Michael Lifsgarden asks, with all the data that you're collecting, have you considered tracking daily diet, either for yourself or in larger cohorts, with the goal of identifying an individualized diet that best optimizes biomarkers that are representative of many organ systems?
Oh yeah, great question. So the answer is yes, we do. I wear a CGM. I've been wearing one for about eight and a half years now. So, and I don't always log diets because it's pretty time consuming, but I've gone through several years of that. And then my company, January AI, does a lot of that too. So we obviously we do find different diets spike different people, and with AI, that's what the company's built around. It's actually able to make predictions of, which foods are going to spike you and which ones won't, based on food compositions and other markers. And we'd like to get markers going into that, something we're working on now, like what profile predicts, even without any data, what you might spike to. We're not there yet. We're working on that, trying to collect that data. So we've done a ton on me. We're doing a whole bunch on other people. We're running people through controlled diets to help make those predictions. There's the last part of that question I forgot that we weren't doing just yet. But anyway, if I remember, I'll tell you anyway. It disappeared from
the optimizing biomarkers representative of many organ systems.
Oh yeah, I don't know if we have all the organ systems pinned down. That's the part we don't know. So we have some, right? We can follow renal. We have some very interesting markers for kidney and some of the other things. And there's a lot of standard markers for those, but we need more for all the organ systems. This is a great thing, especially around the agenotype. I think a lot of the agenotype markers will turn out to be useful biomarkers. So great question. All right.
Chao Kang Lei asks, can precision nutrition be accomplished without human genome information, as acquiring omics information requires so much omics profiling at a high cost?
So the answer is yes, we think so, because we can follow patterns, we can make predictions around people's food they eat, which is one of the major sources of what's going to affect people how. I think having the genome will help us in our predictive model. By the way, the genome sequences will be dirt cheap soon. There's new technology, you may know, for $100 for that. They estimate the cost of sequencing the genome will be $100. Probably interpretation will still be a lot more, but we will get to very, very good numbers for all this stuff. So the answer is absolutely, and that's what we're trying to do, build personalized AI model. I didn't show you, but we have a AI model for me where I can see, it's very easy to see when I'm shifting off my norm in a variety of different ways. So that I think that's the future.
Thank you. The next question, Larissa Constantinou asks, what useful information can urine microbiome provide?
Yeah, I don't know. We haven't done much with the urine microbiome. It's a bit controversial. A lot of people say, well, your urine is really sterile. I think the data now says it's probably not completely sterile. We just don't have much data on the urine microbiome. It's we collected 5, but it's the one of the 5 we haven't analyzed. So good question. I don't know the answer.
Brent Cochran asks, how do you avoid the paradox of overscreening, where things like benign tumors get overtreated, possibly causing harm, like in prostate and thyroid cancer?
Yeah, so I'm a believer you can't have too much information. People worry that we are getting too much, and people leads to hypochondriacs and over treatments. I think what our stuff is just deep screening, and then from that you should do appropriate tests. Now, how you use the information, like over treatment, I do think we have to be careful about these things, but these days are pretty good markers, I think, for benign versus aggressive cancers. And so I think, this is, I mean, historically, you're right. There has been a lot of overtreatment. And I would argue that just means we need better markers to distinguish benign versus aggressive. But all we're doing is putting people on the alert. Even if you see something, you're not sure if it's benign or aggressive, just follow its growth. And I think the whole body MRI is a classic example. It turns out if talk to any physician today, 100% of them will tell you you should not get a whole body MRI. I forgot to say that's what QBio does. It does a medical version of what I presented with whole body MRI. And we say that's wrong. You actually do want a whole body. They say don't do it because you're going to have men have nodules in their prostate, women in their ovaries, which is true. 100% of the time, people have nodules. But that's not the point. The point is, do you have any growing nodules? And so by doing the longitudinal measurements, you see if any are growing. If any are growing, you have an issue. And so the name of the game is to know what you have. I have 9 nodules. I know where there are. None of them are growing. And that's useful because if I ever get cancer and they do a whole body MRI, they're not going to know if these new nodules, the nodules they discovered are new, or if they were there to begin with, or are they bigger or what have you. So we would argue knowing what you have is a big deal. And then again, you can use the information accordingly. So that's how I see it.
Thank you. Okay. Richard Alpert says, fascinating. Are any efforts made to translate your findings to clinical practice, particularly for internists and family physicians?
Oh, great question. So the answer is yes and no. In general, no, because our healthcare system is broken. One other way, which is nobody pays to keep you healthy. What I'm doing is pretty expensive. So what you like it's $3,500 to do the QBio, get the whole body MRI. We're going to drive that price down. We know how to do it. So I hope in the future it will be a few $100s to do what I just presented. Then everybody will do it. But right now you have to pay out of pocket because again, no provider or insurer is going to pay to keep you healthy. I think they should. I think you should get an insurance break if you wear a smartwatch or get your genome sequence. That doesn't happen in today's world. But it should. You should get a bonus because you'll take better care of yourself. So I think we need the right incentive structure to bring it into the system. I think in certain areas, you can actually model how it will save money, like for cardiovascular disease, get some of the stroke who goes on long-term disability. That's extremely expensive. So we need to bring it in better ways. We are trying to bring it out, as you know, this YOLO January AI is a way of getting your CGM and actually train people. You ought to pay out of pocket. It's not that expensive. That one's easy, $280. YOLO, IOLO has the thing where you do a metabolic profile at home testing. It's pretty cool for 500 metabolites. So that's how we're going to roll it out. And then we're going to show it has utility. And then I hope everyone in the clinical space adopts this stuff, whether it's through our companies or through their own clinic. It's all great. That's the goal. Transform this space, keep people healthy.
Great. So I'm looking at the time. I think we'll take one more question. So this question was upvoted. This is from Eric Miller. Can you comment on issues of ethics, data privacy, and related topics that are of such concern to many people these days?
Yeah, it's a great question. I have several different answers to this. One flip answer would be to say, privacy, get over it. It doesn't exist. Right? You have cameras on every street corner, etc. You share your credit card, which has a ton of personal information with credit card companies, things like that. So I think that's the flip answer. And we do take it seriously. I mean, we do try to do this. I do think I think having this information is going to benefit people. And I think if it starts getting abused, that's where you need laws to protect people. And that's the purpose of government, to make laws for people to do things for people they can't do for themselves. So we would want to protect people from being abused by this, using such information for bad. But I think it'd be used for a ton of good, and that's my 2 cents there. I still think it's better to get the information, like your credit card. You don't carry around bags of cash because it's very inconvenient. It's better to have a credit card. So I would argue it's better to get your health profile, even if there's some risk of adverse things, if it can help your health.
Okay, great. So our time is up. This is going to conclude the question section and today's seminar. Thank you to our speaker today, and thanks to everyone for attending. And we'll see you all next week.
Thanks for having me. Bye,
Dr.
all.
Snyder, if you want to stay on the...
Well, thank you very much.